Okay, great. We still have people entering into the webinar, but let's go ahead and get started. Welcome everyone to the ACPMP 2024 educational series. Our June webinar, we're very happy to feature Travera and their presentation on some new and very interesting technology, optimizing treatment selection, exploring a new technology to personalize therapy beyond any standard approach. My name is Deborah Shelton. I'm the Executive Director of ACPMP. Again, welcome everyone. So happy to have you here. Just a brief reminder about the ACPMP and our mission. We're a nonprofit organization focused on two things primarily. One, funding and supporting critical research, hopefully to find a cure for appendix cancer sooner rather than later. And two, we also fund and support educational programs for physicians and patient caregivers and families to keep them abreast about this rare cancer. All of this work is made possible solely through the generosity of our donors. We're grateful for each and every dollar and to each and every one of you. I just want to do a brief bit of housekeeping, but before I do that, since we're talking about donations, and this is Walk Week, I wanted Alyssa Strackbine, our operations coordinator, and the point on our walk to give a brief shout out to it. Alyssa?
Yes, thanks, Deborah. Our walk is still going on through this Sunday, June 16th. We're really close to our goal. We're 92%. So you can still register or donate today. You can scan that QR code right there, and we'll also send it out in an email afterwards. But really, any help, like Deborah said, is great. and you'll still get a t-shirt. Might not come in time to walk, but you can still get one. So thank you guys so much.
Thanks, Alyssa. Okay, so really quickly, just some brief housekeeping notes. One, we will be taking up a Q&A session. We do have some questions that were pre-submitted as part of the registration process. We also welcome questions during the presentation, and our presenters will get to as many of those as they can. If you do have a question, please use the Q&A feature at the bottom of your screen. Do not use the chat for this particular platform. Please put your questions through that Q&A feature. Everyone's lines are muted except for the presenters, and that's just to minimize background noise. We have quite a few people attending this webinar. This webinar will be recorded, and so it will be available for those that have registered. A playback will be immediately available, and for everyone else, a recording of this will be put on our webinar, on our website as soon as we can get it up there for everyone to be able to take advantage of the good information provided. And last but not least, we always welcome any and all suggestions for topics and presenters for our education series. So if anyone has a topic that you'd love to see addressed, please do reach out to me. The email address, my email address is right up here, deborah.s@acpmp.org, or you could simply submit it to info@acpmp.org. That's always welcome. Okay, with that, let me introduce our presenters tonight from Travera. Today's main presenter will be Dennis Watson, and Dennis is Travera's Vice President of Business Development and a 16-year veteran of the whole cancer diagnostics environment. Dennis will be joined tonight by his colleagues, Mark Stevens, PhD, Travera's CEO, and Rob Kimmerling, PhD, Travera's Chief Technical Officer. Mark and Rob are two of Travera's co-founders, and they earned their PhDs at MIT, where they worked in the development of the very technology that we're going to be talking about tonight. Mark and Rob hope to be valuable during the Q&A session immediately following Dennis's presentation. So with that, Dennis, I'm going to turn it over to you. Welcome, and thank you, Team Travera. You might.
Sorry, I still had my mute on.
No
We're off to a great start. Thanks very much, Deborah, for that introduction. And myself, Mark, Rob, we're really honored and humbled to be invited to do this presentation with ACPMP. You guys do some great work, and we're excited to be here and share some of the work that we're doing. So I'm going to run through these slides in probably about 30 minutes, and then we'll take some opportunity for questions at the end. So bear with me. Some of this will get a little bit heavy, but I try to stay as top level with it as I possibly can. And as Deborah introduced, this presentation is really about unique ways to optimize treatment selection, and when we get beyond standard of care therapy, so really beyond first line therapies into what comes in second, third, and later lines of therapy in many cases. So we're going to go through a few different things here. We're going to set up kind of the challenge. It's a challenge that many of the people on this call are probably already well aware of, but we're gonna just quickly set up the challenge that Travera is trying to solve. The solution that we have come up with and that we're trying to continue to validate and grow into the market. And then we're going to go into some next steps and Q&A. So first, again, I want to start with what is the problem? And it's a well-known problem in rare disease. It's a well-known problem in late stage disease as well. But the challenge that we're trying to solve is, how do we match cancer patients to the right drug? It's a really difficult problem. And after first line therapy, there's about 270 plus drugs approved by the FDA for different types of cancer treatment and that are listed in the guidelines. But the likelihood of a patient getting matched to a therapy that gives them clinical benefit after first line is less than 25%. There's a lot of people in the cancer space, in the diagnostic space, in the medical space that have been working to improve this number for many, many years. And the number is better than it used to be, but there's obviously still room for improvement. And one of the places that we've seen improvement is through precision medicine. Precision medicine is using things like genomics and genetic testing to help patients get matched to a drug that might be better targeted to maybe a mutation that's been found in their tumor. I personally spent 15 plus years in this environment and in the diagnostic market. And unfortunately, genomics has just not gotten us to where we thought it would. At this point in time, the likelihood of a patient being matched to a drug through genomic sequencing that works for them is less than 15%. And so there's still a real gap that exists in trying to get patients closer to these therapies that might be beneficial for them. And this is where personalized medicine can come in. This is a phrase you're probably all familiar with. It's a phrase that is often used in medical practice today. But the idea of personalized medicine is really matching the therapy to the patient in their unique circumstances as directly as possible. And one way that we do this in cancer is with a type of testing called functional testing. And the idea of functional testing is selecting the best drug by testing the patient's live cancer cells. So this is actually not a new idea. Travera is approaching an area of oncologic science that has been approached before. There's a long history of testing drugs outside of the body against cancer cells. The problem is that historically, this kind of technology and this kind of approach has been slow. It can take weeks or sometimes even months to accomplish. There's limited breadth across different types of cancers, different types of samples, and different drugs. And historically, it's required us to grow cells. And that is a really important caveat to understand. The reason it's important is because the process works a little bit like this. To be able to test a drug in an artificial environment against a living cancer cell, that cell has to be alive, which means it has to be collected from a biopsy or a fluid drain or somewhere where we can get access to living cancer cells. And we then have to try to see whether or not the drugs are going to kill those cells in this sort of created environment. The problem is that when you take a cell out of the body, you remove it from its life source, it only will live 3 to 4 days on average, maybe less. But the average cancer therapy drug can take a week or more to work. And so we have this sort of time conundrum where the cells are dying faster than the drugs can kill them. And so how do we measure a drug's ability to kill a cell if the cell's going to die faster all by itself? We've done it in the past by intervening. So we get those cells and we grow them. We grow them in cultures like many of us did in 9th grade biology class with bacteria in a petri dish. We grow them into mice. We grow them into more recently organoids. And there's a lot of different technologies that have improved dramatically over the years to get us closer to duplicating what's happening in the patient's body. The problem, though, with all of this is that cancer is a mutant. And so when you take a cancer cell out of the body and you pump it full of growth factors and proteins and all the things you need to feed it to get it to grow, the cancer often changes. And so we end up testing drug here against something that isn't really what was directly removed from the patient here. We've gotten closer, but there still is a gap that exists there. And this is one of the reasons this kind of approach hasn't been widely taken on in oncologic treatments. Travera's approach, however, is a little bit different. It's actually a biophysical approach. We weigh the cells, and it's as simple as it sounds. It turns out that single cell mass works as a nearly universal biomarker. And this is what that looks like. When you take a cancer cell out of the body, like I said, and kill it using drug or otherwise, cancer cells lose about half of their mass, half of their weight in death. And that process generally takes a few days. While cancer drugs can take the days to kill the cancer cells, an effective drug will first begin changing the cell on its way to killing it. And if you have a sensitive enough tool to measure that weight change, you can see this in almost real time, often in less than 24 hours. And that's really what Travera is working and has already been doing. It's all because of a new invention that came out of MIT. And I'm not going to go into this. There's a lot of numbers and words on this screen. But I like the animation and I want to share with you. But the real device is this little chip that's on this fingertip here. And essentially, the way that this works is we pass single cells through that tiny little chip, and it's blown up on the right side of the screen here. And if you can imagine that little red dot is a cell, as we pass cells through this chip, they go through this little channel and they pass over the end of that thing that looks like a diving board. And it's very similar to a diving board in that when you jump off of a diving board into a swimming pool, the diving board vibrates. It turns out when cancer cells pass over the end of this little cantilever, the cantilever also vibrates. And the frequency of that vibration directly correlates to how much the cell weighs. At the end of the day, what this really means is we've devised a tool that can weigh individual cancer cells at a rate 100 times more sensitive than the previous existing tools that are out there today. And so this is how all this kind of translates. This is what you've learned so far in this presentation, I hope. Number one, an effective cancer therapy changes a cell before it kills the cell. Number two, just as your body weight changes and is measurable on a scale if you lose or gain fat or muscle mass, the same thing happens in a cell. As the cell structure is impacted by the drug, that cell structure changes in a lot of different ways depending on the drug, but something changes in that cell. And those changes that are occurring in the cell translate to a change of weight. And Travera now has the first tool that is sensitive enough to recognize those tiny alterations in cell mass. And so this is really the technology that makes what we do possible. And this is how it works in the clinic. We have to start, just like with the old generation things that I discussed, with live cells. So it starts with a live, fresh biopsy. Can't use biopsies that happened 3 months ago, 3 weeks ago, or even 3 days ago. We have to be able to have our specialized test kits on hand so that when the cells are collected through a biopsy, a fluid drain, or other methods I'll go into in a little bit, those cells can be immediately placed into our test kit and delivered FedEx overnight to our lab the next day. Once we get those samples in our lab, we're going to move with that specimen, and we're going to pull out all the cancer cells we can get out of the sample that we get. We're going to put them in about 5,000 cells per well into this little plate here called a 96 well plate. It's basically just a bunch of different cups. And then each of those cups are going to get a different drug. Some of those cups are also going to not get any drug at all. So we have drug cells and we have control cells. The idea being that all of these live cells are slowly changing since they've been removed from the body. But if the cells with drug are changing faster than the cells without drug, then that's what tells us these drugs are beginning to work in these cells. If the drug cells are changing at the same rate as the control cells, it's almost as if it didn't matter if a drug was present or not. And so the concept being that drug was basically a placebo. It's not having an effect on these cells. And that is really conceptually how this works. So you send in a cell specimen through a handful of different methodologies. We get the cells out of it. We test different drugs against those cells. And for the first time in the history of testing cancer cells outside of the human body, we have a device that allows us to do it without growing cells, which means there's less of a risk of those cells mutating and changing and becoming different over time. We're able to test real drugs against the direct cells that were removed from the patient, and we're able to do it very rapidly. Once we complete that process, we return a report to your doctor. We'll run up to 20 drugs at a time, and we'll return a report to your doctor that looks something like this. It's going to rank all of those cells, all of those drugs, based on how they affected the cells. The more of a response we saw in the cell, the higher it goes on the chart, and the more sensitive those cells appeared to be to that drug. And the less of a change we see in those cells compared to the baseline, the lower those drugs fall on the chart, showing us drugs that are less likely to elicit a response. We score these right now on a scale of 0 to 100, but as we continue to build out our data, where we are with the data today is really at a place to say, this is a binary result for the most part. If a drug scores above 50, it has a higher likelihood of creating a clinical response. If a drug falls below 50, it has a lower likelihood of that. And so the concept of all this being, you've been through first-line therapy, maybe you've been through second-line therapy, you're at a place now where you're trying to decide what to do next, and there's a list of available drugs in front of you. The selection process of what drug to try becomes an arduous one. It's often a trial and error approach. It's based on a lot of different information the physician has available to feed that data. But at the end of the day, it's let's try this. We'll monitor you closely. If it works, we'll keep going. If it doesn't, we'll try something else. The concept that we're laying out here is let's look at the response of the cells to these drugs before we put these drugs in your body. And that's really what the intent of this assay is. So right now we've been able to do this on a wide variety of cancer types. You might notice that appendix cancer is not listed in this chart. That's actually only because this particular list of 39 cancers is just the list of cancers we have predetermined drug lists for. With appendix cancer, because it's a rare disease and because the available drugs are more limited, we work with the clinicians on an individual basis. We have more than 100 FDA approved drugs that is available for us to analyze. Like I said, we attempt to test as many as 20 drugs per specimen that we receive. And the idea being that we'll give the readings back on those 20 drugs. We prefer usually to start and end with drugs that are indicated for the disease that we're testing. But in rare disease and later stage disease, it is not at all uncommon that some of these FDA approved drugs are maybe used off-label. And so working with your physician will create really a customized list that says, these are the drugs we want to test for this patient because we know what they've had before. We don't want to repeat those. And we know what we're interested in looking at moving forward. So at the end, kind of the next step of this is how good are we at bringing this out of the clinic and really showing that it works. So keep in mind that I started out this presentation sharing that the chance of response for pre-treated patients in second line and later therapies is about 25% or less. And I believe it is smaller in rare disease. But at this point, we've tested a little over 800 clinical samples in through our lab. And we have full outcomes on about 70, a little over 70, 75 of those patients today. And our early predictive data suggests that Travera triples the chance of selecting an effective drug in each of these cases. This means that These patients that you see listed in this box in the far right are patients that received the test. We ran a panel of drugs. Their clinician then gave them one of those drugs that we either said was going to work or we suggested would not work. And 76% of the time, we have accurately predicted whether or not that drug was going to elicit a response in that patient. And so this is data that we are excited about, but it is data that is early. The numbers here are small, as you can probably see. And so we are continuing to work through more clinical trials, more validation efforts, and really ongoing development to continue to improve, enhance, and move on from the data that we have today to really bring this to the masses and as clean a way as we possibly can. So let's shift a little bit and focus on appendix cancer specifically here. As a rare disease, appendix cancer is something that we don't have as much experience with as we do some other tumor types, but we do have some. And it's an area that we have actually very specifically focused on increasing our efforts in. We're working with a lot of clinicians that specialize in appendix cancer around the country at a lot of different academic institutions. And we do have some data to speak to appendix cancer specifically, and more focused on whether or not we're able to get a successful test when we run an appendix cancer sample. So this is what that looks like so far. We have done 13 cases of appendix cancer in the last 3 to 6 months. And so far, we have produced reports with scored drugs on 6 of those 13 cases, which means 7 of the samples failed to produce a report. And we weren't able to test any drugs or analyze any drugs in those cases. Now, I want to add some additional color to that because the numbers aren't quite as bad as they look. Among those 7 cases that failed, one of them got to pathology and did not have any active disease. Pathology wasn't able to identify cancer. We weren't able to identify cancer. Without cancer, we don't have anything to test drug against. And so this was really a patient that probably good news for that patient, didn't have active disease in the sample that we received. There are another 3 patients out of these 7 that at the time we received their sample, they were receiving active chemotherapy. They had gotten doses of chemo within a week or 2 of the biopsy being performed. The reason this is relevant is because keeping in mind, we need live cancer cells. And so even if the patient is on an active drug that doesn't seem to be working great, it might still be working enough that it weakens those cells to the point that by the time those cells are biopsied, put into the test kit and shipped overnight and analyzed in our lab, that's a lot of wear and tear on cells that have already been weakened by a therapy. And so the likelihood of success in those cases is lower. And so if we take out the patients that were in active treatment and the patients, the one patient that turned out not to have identifiable disease, then we're really left with a success rate of about 6 out of 9 or 67%, which isn't bad for small numbers. And we do hope to continue to improve on this. One of the other things, however, that's important to understand is while we do try to target 20 drugs per specimen that we analyze, we're limited by the number of cells that exist in the sample that we receive. And appendix cancer specifically is known for being difficult to isolate cells from. In these 6 samples, we did isolate cells successfully, but we didn't have enough cells to run all 20 drugs in most of these cases. Most of the cases that we've done to date in appendix cancer, we ran about 5 drugs. And so it's important to understand that when we're working with you and your doctor on what drugs we want to run against the specimen, we're going to come up with a list of 20. We'll always give you a list to start with and allow you to edit it against that larger list of available drugs. But working from that list of 20 drugs, we also like to prioritize what's most important. to you and the physician. If I get in a situation where we can only run 4 or 5 drugs, I don't want to run the 4 or 5 drugs you've previously had. So I keep saying isolation, and I want to give you just a really brief explanation as to what that means, because it's an important thing to understand when you think about the fact that we need to get access to a certain number of cells. So I'm going to give you a very quick biology lesson. I'm not going to go into the details of the things that you see on the screen here, but generally, I think when we think of a cancerous mass, whether it's a liver metastasis or a peritoneal metastasis or a primary tumor, we think of just a mass of a bunch of cancer cells. In actuality, there's a lot of other stuff that's in there. There are immune cells, there are blood cells, there's stroma, which is kind of connective tissue, and other things that kind of give that mass its structure. There's a lot of things that are present. And so when the doctor sticks their needle in to take this specimen and they come right down through here, they're going to get some of those light blue cancer cells, but they're also going to get any of these other cells that are just in the line of that needle. And so it's important to understand that when we're isolating those cells, the first thing we're going to do with your specimen in our lab is work to get rid of all that background stuff so we can just focus on the cancer cells. And so you might say, but my doctor sent you a good sample. There's a great sample here. I know we got a nice chunk of tissue. That might absolutely be true. But the good sample is just step one. Once we get that sample, we need to then be able to pull out that background stuff and see what we're left with. And because the ratio of how much stroma or immune cells or other things that are in that particular tumor varies so much from patient to patient and tumor to tumor, there's really no way for anyone to know how many cells we're going to be able to extract until we get that specimen in our lab. And so we're going to do the best we can with the sample that we get, but in some cases, we're left with a specimen that just doesn't have a lot of cancer cells in it. And so we might then have to run those limited drug lists like I was talking about earlier, or in some cases, we just might not have enough cells to test at all. Or in the cases I described where the patient's on active treatment, we might have a situation where the cells that we have are no longer alive, which creates obvious difficulties in trying to run a live cell test. In appendix cancer specifically, there's a couple of things that can also complicate this. When patients are heavily pretreated with appendix cancer, it is not uncommon that those tumors increase stroma. And so we end up with more stroma or kind of connective tissues and other tissues in the mass than we do cancer cells. We've certainly had plenty of successful cases, but it is something to just be aware of if there have been a lot of therapies received in the past. The other thing is mucinous histology is common among appendix cancer. And as you can imagine, in that tiny little chip that I showed you earlier, and those little channels we're passing cancer cells through, a cell that's creating mucus as it goes can gum up the works, for lack of a better word. And so we have a lot of little tricks to try to prevent and reduce that, but mucinous histology does also slightly limit the success rate of getting a good sample when we're dealing with appendix cancer. So what's an ideal case? What's kind of the perfect scenario? And there's really 3 points to be thoughtful of. The first is that you're planning to change therapy or initiate new therapy soon. Because cancer changes over time or can change over time, the idea of having a biopsy done today to run the Travera test and tell you how predictive, how sensitive your cells might be to different drugs, and then not starting those drugs for another year might mean that the cancer you're treating those drugs with a year from now isn't really representative of what it was when we first sampled it. And so the timing of trying to get the sample close to a decision point in therapy change can be an important one. That said, we're not getting biopsies and fluid drains and all of these things done every single day. And so we also want to be somewhat opportunistic. If there's an opportunity to get access to live cells, which we need to be able to run the test, then we're happy to run the test. We're happy to run the test even if you're in active therapy. We're happy to run the test if you don't plan to make a change in the next few weeks and it might be a little further down the road. We're happy to sort of be opportunistic and take advantage of the sample that exists. We just like to be really transparent on the benefits and opportunities of these different kinds of approaches. So again, we do need those live cancer cells. We can do this from malignant fluid. If you have ascites, that acidic fluid tends to work really well in the process. And so if you're going in for a paracentesis, or if you have a Prolix catheter to drain that fluid from, we can get the specimens that way. Or if you're going in really for any kind of surgical procedure, biopsy, diagnostic laparoscopy, these are all things that have yielded really nice samples for us that have worked well. But again, we cannot use archived tissue, frozen or otherwise. Finally, we are a CLIA approved test. That means we are approved for clinical use in the United States, but it does have to be ordered by a treating physician. This is not a test that a patient can order for themselves. And so often when patients become interested, they begin with us and we help them in working through that process with their doctor. There's one other additional note down here at the bottom that we are not currently available in the state of New York. That CLIA approval process, New York has a slightly different process that just takes longer. We're deep into it. It's been going well. But at this point, we are still just waiting on the final approval notification from New York. However, in the meantime, New York has a great appeal process that we filed with them many times, and it tends to work well. We've not had one be denied yet. We just need a couple of extra days notice to be able to work through that appeal process with you. We also have clinical trials that you can enroll in that allow for samples to be submitted from New York. So there are some opportunities to still work around that while we're waiting on that final approval to come through. So finally, what do next steps look like? There's just a couple of slides left, I promise. You know, the first thing that I want to make sure everybody leaves this understanding is that Travera is early. Our data is early. It is exciting, but it is still developing. And as we continue through that development, we want to partner with both patients and physicians to continue to improve and enhance our assay. And so right now, we are offering our test at no cost. Working with groups like ACPMP and others, being able to offer the test at no cost creates a really nice environment for both of us, and that you and your doctor get access to this developing early technology and the exciting data that's behind it. And Travera gets to learn from each and every case we run. We get to continue to improve our process. We get to continue to expand on our data for not only current patients, but future patients. Please just note that Travera cannot pay for a biopsy or procedure. And so while we will run the test for free, we can't do the biopsy or procedure for free. So we find it's best just to kind of coordinate with collections that are already planned as part of your normal care. As I said earlier, diagnostic laparoscopies, pleural effusion or ascites drains, things that come up throughout the normal clinical care environment that are going to give your physician an opportunity to access live cells. And we simply coordinate with the timing of those procedures to make sure we can get a small portion of that sample. And so if you're interested in moving into this and trying to find out how this test might apply to you personally or individually, the best thing to do is to just reach out. You can go to travera.com/contact. And you can fill out the contact form that's there, give us a little bit of insight on your case, and I will actually directly follow up with you to answer any questions you may have and kind of explain what the logistical process sort of looks like, work with you to help communicate with your physician, etc. And once you have the information that you need through that process, you can contact your doctor and share about this new test, your interest in having it, and the different opportunities available to get it. Finally, once all that happens, the real process, and this is pretty much the last slide, is once your doctor is on board and supportive with doing this assay, they complete the order form. We send out a test kit. Because we're doing a lot of N of 1 cases with institutions around the country, we often will send the test kit directly to the patient, just because that lowers the risk that it gets just misplaced in the hospital. We mail the kit to you, you take the kit with you the day of your biopsy, your procedure, whatever it might be. And then the sample's collected by the physician, and then we coordinate a way to get that to FedEx that afternoon or following that procedure to get it to us then next day. Once we get that tissue and sample into our lab, we run through that process that I briefly described earlier against the drugs that your clinician and you have identified from the list that we have available. And then about 2 days later, we'll return the results of what we found in the relative sensitivity of those cells to those drugs. Then at some future point, we're going to reach out to you and ask about your experience, potentially ask you to enroll in our registry, just find ways for us to get access to what happened in your case. So like I said, we can continue to learn from that and improve in that through both patient and physician directed partnerships. The last thing I'll leave you with is I just want to make everybody here aware that Travera is newer to the appendix cancer environment. We're continuing to expand our data there, but we are in the early stages of kicking off some really expanded efforts in appendix cancer, specifically along with other GI-based malignancies. And we're working to further validate our assay in those areas, working with a lot of major appendix specialists and academic cancer centers around the country. We've got some really nice partnerships and that are developing to do that. And we're focused on continuing to validate the predictive accuracy of our test as a biomarker, not only in traditional therapies, but in high-pack based approaches as well. The idea of being able to have a 2-day turnaround time on a live tissue specimen that could give a surgical oncologist an indication as to whether or not this particular high-pack based approach is going to work in this particular patient could be a really valuable tool. These are early hopes and dreams, but there's a lot of data to set up the promise of what we think we're going to be able to do there. So that is the end of the presentation. As I said, you can contact me. This is my personal email address, or you can go to our Contact Us page if you would like more information on whether or not testing might apply to you and how you might go about getting it. And so if Rob and Mark would like to join me on camera here and unmute, we did get a handful of pre-submitted questions, ran through just a quick selection of those here, and then we'll go to the questions that have come on since the opening of the webinar as well. So I'll take these first couple, and then maybe Mark and Rob, I'll pass them off to you. First question is about access to clinical trials at AHN in Pittsburgh. We don't have any clinical trials right now running at AHN that are specific to AHN. However, we do have this registry trial that I briefly mentioned earlier that is that's a really nice trial design because it's sort of an observational registry that is done in a decentralized manner, which means patients can individually enroll in this from anywhere in the country. And it's about a 10 to 15 minute phone call to get consented. And we usually connect patients to that after we know there's a viable report from the procedure. And the main purpose of that study is really just to track that outcomes data that's going to be important for us to continue our development. Can samples previously biopsy be used? I certainly hit on this during the presentation, but just for the sake of clarity, we cannot use old archived or frozen specimens. We need fresh, living, alive cells to be able to see how those cells respond to those drugs.
Yeah, and when it comes to the next question, can this process be used to determine if a currently prescribed cancer treatment is actually working? The process that Dennis described today is really about treatment selection and not about treatment response. I will say that we did produce data in our prior academic lives showing that this mass change also can be observed from responding tumor cells in a patient, but that's not something that Travera is working on. So that's not the function of what we're doing today, although perhaps someday that's a way we'll be able to use this tool. When it comes to the next question, could this be valuable to a patient with a high-grade signet ring adenocarcinoma after CRS HIPEC? That is really subject to whether or not there's still accessible tissue after those procedures and how long it's been since those procedures took place. So Dennis spoke about this, but if we're within 7 days of ending a round of chemotherapy, we're still unlikely to get a usable tumor sample. And it is also a decision for you and your doctor as to whether the tumor itself is biopsiable by one method or another. And so that's a conversation we're happy to help with, but is subject to those constraints. When there's this question, do you have any options for patients with LAMN, a subtype of the cancers? And you know, this is something we're happy to talk about. Again, we do custom panels of drugs, and we are receptive to working with oncologists to make sure those panels fit your disease. If it's something we have less experience with, though, you know, we do want to set expectations that much like we are early in our data production as a whole, if it's truly a sample type we've never dealt with before, you know, we do want to set expectations there that it might be more challenging. But so far, we've worked well with a large range of different types of carcinomas. So, and Dennis, do you want to take these next ones, or Rob?
So I can take the
Yeah.
question of on the chemo side. As Dennis mentioned, we do have a pretty large panel of validated drugs at this point. So it's beyond just the sort of standard cytotoxic chemotherapies that are very common. There's also a fair number of targeted inhibitors that we're able to test. So this is something we can work in sort of consultation with your treating oncologist to see what the priority list of those drugs look like based on which of the drugs we have validated on our end. But we're happy to customize that panel as Dennis mentioned.
The final question that was pre-submitted is this testing offered to patients in Canada? We do test patients from Canada quite regularly, and really anywhere in the world that has FedEx overnight delivery available to our lab in Boston. And so that tends to be Canada, Mexico, and sort of much of Central America, as well as most of Europe. And so as long as we can get tissue available to our lab overnight, it's not a problem. I noticed in the Q&A, there was a question about legal issues and transporting live tissue across the border. And there is, of course, a customs process that has to be completed. But we've done this numerous times without any real challenges. So maybe we could go on to some of the questions that have been submitted. And I think I'm going to let Rob take this first one. I'll read through it real quick. But when carrying out the analysis, how do you determine efficacy of drugs such as checkpoint inhibitors, which are codependent on an immune response? And this is an important question. I didn't go into this during the presentation, but we do have a checkpoint inhibitor panel, but it requires access to immune cells, and it's not something that we do from tissue. We can do it from right now just from malignant fluid like ascites. But I'll let Rob touch a little more in a little more detail on kind of how that assay works compared to the assay that I described?
Yeah, so this is a slightly different technical approach. The platform that we're using is the same in that circumstance. We're looking at the weight change of cells, but instead of looking at tumor cells, we're actually tracking the weight change of immune cells, so T cells in this case. So as Dennis mentioned, this requires a specimen that has access to immune cells. We treat with activation and those checkpoint inhibitors ex vivo in our lab. And then we look at the weight change of those T cells as a marker of whether or not those drugs are inducing more of a rigorous T cell response. So that's how we are currently functionally reading out the checkpoint drugs so far. And it's a slightly different flavor of the assay, but it's something that we do run pretty frequently as well.
The next question here, I think we sort of already addressed in the pre-submitted questions. So I'm just gonna skip that one, and if whomever submitted that has more questions about it, you can feel free to email us, and we're happy to answer it one off as well. Next question, you mentioned that the live cells are sent via FedEx overnight to the lab. Oh, this is that Canadian tissue question. So we already addressed that as well. Are you approved for working with patients in Canada? That is a yes. So we will answer that one and move on to the next. Are you doing any clinical trials for patients in Canada? So we do not have any clinical trials open right now in Canada. The trial that I mentioned earlier, the registry that's pretty easily to enroll from anywhere in the US, is unfortunately not available in Canada. However, the point that I mentioned earlier, that we're running these tests at this point in time at no cost, and our ask in return is that you let us know what happens. We're happy to sort of work out that same kind of understanding with the Canadian patient. It just means that it won't be done through a formal trial. That's still really valuable feedback for our ongoing development of both our test and our data. So we're happy to work with patients in Canada. Besides FOLFOX, what are some of the other drugs you're testing for appendiceal cancer? So as I said earlier, we have a list of more than 100 FDA cleared, FDA approved drugs across all cancer types. And so, many of those drugs might be specific to leukemias, for example. But we have a really nice list of both chemotherapies as well as targeted inhibitors that can be valuable for analyzing against cells in appendiceal cancer. Most of the cases we've done to date in working with the clinicians directly, we often will start with our predetermined colon cancer panel, which is 20 drugs indicated for colon cancer. And that's just sort of the starting point for the clinicians to say, hey, here's a list. It's easier to edit this list than it is to create a whole new list. And so that's FOLFOX, it's FOLFIRI. Because of the prevalence of things like HIPEC, we often will do mitomycin or cisplatin in these patients. But there's a really long list, and we like to work with the clinician and really personalizing it to the individual patient and their scenario. Next question. If you are on chemo, should you go off for the test, and for how long? So that's a tough question because there's a lot of different variables at work there. As I said earlier, our technology, the data is exciting, and it's very, very promising, but it is still early. And sometimes these cases don't work, like I reviewed during the presentation. We don't get enough cells. The cells we get are damaged, or there's too much other stuff present in the sample. All of these different variables that we just can't ensure that every sample we are going to get is going to definitely produce a good report that gives you exactly what you were hoping to see. And so understanding that variable and kind of the risk that exists there, we don't usually recommend scheduling a biopsy specifically for the reason of getting our test done. And the other reason we don't recommend scheduling a biopsy for our test is while we're giving the test away right now at no cost, so we can capture data and experience, our test is not currently reimbursed by insurance. And so because the test isn't reimbursed by insurance, a biopsy ordered specifically for the test wouldn't be reimbursed by insurance. And so we don't want to put you in a position where you have to pay out of pocket for a biopsy when there are many different clinical reasons that biopsies or fluid drains come up. And so the idea of stopping your chemotherapy for a couple of weeks specifically to get this test done, I wouldn't recommend. But to answer the question in a slightly different way, we really recommend kind of we call it a washout period or a period without therapy of at least 3 to 4 weeks, ideally, when you're actively receiving chemo. So if you're between treatments, you're thinking about changing treatments, and you've got a 3 to 4 week window, maybe you're getting a diagnostic laparoscopy done, those are the kinds of things we just try to take sort of advantage of at the time.
I think one other point there, and on the question about drugs that's worth noting is, we really view ourselves as collaborators with your oncologist. So when it comes to these decisions that affect your care, that's a conversation you should both have with your oncologist and that we're happy to have with them as well. That's really how we like to handle things at this stage because we're so early and because there's a lot of other important information going into your care that has a lot of evidence behind it as well. And we don't take that change in decision-making process lightly. So definitely make sure that your oncologist is involved in these sorts of discussions.
Rob, you want to take this next question about sample options beyond laparoscopy? Rob has done a lot of work in our research and development and works with the NCI and other groups. So I think you might be great to answer this question.
Yeah, for sure. So as Dennis mentioned, our main constraint is we need access to live tumor cells. And so for peritoneal disease, often a laparoscopy is a really good option for that, mostly because these are happening frequently as part of standard of care, like Dennis mentioned. So we're able to get access to that tissue fairly often, which is helpful. I think there are other methods, including core biopsies, that we're also able to work with if we have enough tissue. And then, as mentioned, Dennis also mentioned earlier, fluid access. So if there is any malignant ascites that are developing, if those are being drained, we're able to get cells from that fluid as well. So it's definitely on a case-by-case basis. We have the protocols to work with many of these different tissue formats, including fine needle aspirates, core biopsies, resections, the laparoscopic tissue samples, and the fluids. So again, it's really based on the patient and what they have available as part of their clinical process of actually getting these collections done. So we're happy to work with whatever's available.
Great. Who pays for the drugs? Great question. So that's something that, the doses that we're using in our lab, we're dosing very small volumes of cells. And so the doses that we are using in our lab are just minuscule in comparison to, what a typical use of these drugs would be and compared to what they would be putting in your body. And, they've been adjusted to the volume of cells that we're testing. And so the drugs that we have in that list of available drugs, chemos, targeted inhibitors, et cetera, are all things that we have as part of our lab. It's part of our flow. And we don't charge per drug in that way. Now, if this question is more directed to once we suggest or indicate that a certain drug might work, who then pays for the administration of that drug, that is something that's going to, come up to you and your oncologist and your health insurance and your medical coverage, et cetera. That's not really something that we get involved in. It's one of the reasons that we prefer to start our drug lists with drugs that are already approved and indicated for a certain disease state. Because while it's not uncommon to use drugs off label, especially in rare disease, using a drug off label can create reimbursement challenges. But a lot of clinicians are pretty well versed at figuring that out and working with you on that. So I'm hearing you say that running the Travera test at the time of active disease recurrence and confirmation in conjunction with determining our next course of action for treatment. So we hang back with our NED biomarker CT scan testing until something actually needs to be done rather than getting ahead of it. We need live cancer cells to be able to run the test. And so if a patient is in a situation that they have no evidence of disease, there isn't any live cancer cells to be able to collect and send. While things like biomarker tests, other types of biomarker testing, genomic testing and things like that can be run off of archived tissue, the main idea of this is, how do these cells today respond to these drugs today? And that can change over time. That can change in response to different treatments. That can change in a lot of different ways, or it could not change. But at the end of the day, the best way to use the tool is when you're looking to initiate or change therapy. Are you publishing results or planning for PMP patients? So we don't have any experience with PMP to date. And Rob, I don't know if you or Mark might be able to answer this question any more specifically, but we're primarily indicated for carcinomas. And I don't know that PMP even technically would be classified as a carcinoma, but I'm a little bit out of my depth on that one. So I'll see if one of these guys have any more insight.
Yeah, we unfortunately don't have any experience with PMP just yet. But in terms of publications, we do have our clinical publications and our workflow publications on our website. This is something we care about a lot, is publishing the results and sort of getting out in the research community. We just presented at ASCO as well. And so all of that is available, and we're happy to share whatever data would be valuable or people are interested in. So that's something, as a policy, we're very open and active with publishing. We just don't have results for PMP just yet.
Are there risks in collecting the sampling of cancer spreading? I don't know the data on that. I've heard this many times over the years in my career, that having surgery or having a biopsy creates an opportunity for spread. I don't know the data on that. I can't speak to it directly. But again, we typically don't recommend getting a procedure biopsy done specifically for this test. We typically recommend just taking advantage of other clinically indicated procedures that are already being planned, and then just being, opportunistic with planning and coordinating our sample collection around that. If you have suspected appendix cancer due to all of your tumor markers and tests pointing in that direction, but your disease cannot be officially categorized in this area, is the testing still available? It is. We often run tests for diseases of unknown primary. It's not at all uncommon, as I'm sure many patients and caregivers on the call have maybe experienced or heard about. But it's not at all uncommon that appendix cancer is often identified as something else initially. And so, yeah, that is absolutely something we can still do. what it might change is what drugs are analyzed. So again, just working closely with your doctor to understand that process, understand how they're looking and hoping to treat you, what their plans are. And, you know, we're more than happy to help out any way we can and, you know, running any of those types of specimens. Sorry, I'm just kind of sorting through the questions here. Looks like we've got a couple questions left. A lot of stage 4 appendix cancer patients have been told chemo won't work, and so doctors say there is nothing else to do. So what I am hearing is that Travera might not be best for these patients. That is a difficult question to answer. You know, so obviously it is a decision of you and your doctor as to whether or not continued treatment makes sense for you. And there's a lot of different variables that come in to that decision-making process. You know, but if your clinician is interested in exploring developing technology to see if the cells are responsive to different candidate drugs that they might be thinking about, then we're happy to run those specimens and provide what feedback we can based on our analysis. And final question, would you be able to test the response of drugs in development, peptides, one such examples, Professor Flatmark's GS alpha peptide vaccine? So right now we are not testing drugs that are not FDA approved. We're not doing any investigational drugs. Theoretically, they could be done on our platform, but there's a validation process that we have to go through to understand how the drug is going to work in different cells. You know, there's a lot of different tests that we have to do before we validate a drug on our platform to ensure that the dosing is right, for example, and you know, different things like that. And given that we're a startup, that we're focused on expanding our existing validation efforts and data, we're primarily focused on the drugs that are being most commonly used and that are already FDA approved.
Dennis,
Mark, Rob, would you add anything to that?
Dennis, I'm sorry to interrupt, but just on that point, because there's often confusion, do you want to just repeat the information about when we talk about FDA approved drugs, we mean for other indications. We recognize in this case, not for appendix cancer, but the whole off-label use concept often used to treat cancer. Because I don't want people to hear, oh, we're only testing FDA approved drugs and think, well, what's the point? You don't have any for appendix. Yeah.
No, totally understand. Yeah. No, when I say FDA approved, I mean drugs that have been approved for use by the FDA to treat cancer. A wide variety of cancers. We have over 100 approved drugs on our platform, but they're all drugs that have an FDA indication somewhere. You know, they're not drugs that are just purely investigational molecules, for example. Yeah, no, thank you for adding that clarity, Deborah.
No, thank you. And it looks like, I think, am I right? Did you get through all the questions in the Q&A?
I believe we did.
Very impressive team. And for that last questioner, just so everyone knows, ACPMP has funded one of the funders of Dr. Flatmark's research for the clinical trial. She hopes to get going on that GNAS mutated peptide vaccine. So ACPMP is working and following Dr. Flatmark's work there very carefully and very excited about that. Okay, well, you all have stayed over. You've been very generous with your time. Really appreciate all three of you. Thank you, Team Travera. I think this was a really excellent presentation, highly informative, very comprehensible. So thank you. And I think with that, we'll just close out. Yeah, so
Great.
thank you very much. And thank you to all of our participants. Thank you for all of the great questions. And we look forward to seeing you next time for our next webinar. Stay
Thank you
tuned
for
for
having
that.
us.
Thank you.
Thank
Thank you,
you.
Dennis, Mark, Rob. Okay. Good night, all.
Night.